Now we talk about third generation sequencing machines, and these machines claim to correct the issues which are posed by second or next generation sequencing machines. So for example, they claim that there are no PCR issues since they do sequencing of the single molecules. They don't amplify the molecules. Since the phasing and dephasing steps are also removed, so there is no more putting the chemicals and removing the chemicals. So in this way, they consume less chemicals. They also claim to produce longer reads and better quality, and also claim to have higher throughput. Three important platforms which we put them in third generation sequencing. One of them is actually a history lesson which is actually gone, but we still keep it in order to just look into its algorithms and how it works, Helicos. Then we have Pacific Biosciences, and we will cover Oxford Nanopore. Helicos was a publicly traded company till 2012. It was established at Cambridge, Massachusetts, but it was delisted from NASDAQ in 2010 due to bad performance in terms of managing the finances. It was co-founded by Stanley Lapidus, Stephen Quack, and Norbert Effian in Cambridge. Here was their machine, that is actually Helicos Genetic Analysis System, or GA system. So in third generation sequencing, there is just a library preparation and then there is sequencing. So there is no more amplification or PCR involved. Helicos GA system produces reads which were 35 base pairs. Throughput was 35 GB. And total reads produced per run, they were 600 to 1000 MBs. And the accuracy as per their claims was 97%, and its total runtime was 8 days. Pacific Biosciences have their machine known as PassBio RS. This company was founded by in 2004 at Menlo Park, USA, California. It uses SMART, single molecule real-time sequencing. That is the website if you guys want to look into it. This is PassBio RS machine. Read length it produces was 3000 to 1500 base pairs, so quite longer reads. Throughput was 1 GB, and total read it produces were 70,000. And accuracy as per their claim was 95%. And the total runtime was 30 minutes, so quite quicker. Oxford Nanopore, actually that technology was established at Oxford University, UK, and then since 2005 they are trying to do the sequencing in quick speed of time, and where they claim that probably they are producing the quickest machines and small size machines. If they are successful, obviously these machines will make a revolution in the genomic technologies, but still they are in the phase of development. So far they have raised over 145 million pounds in investments. These are two machines, Minion, a small USB-like, as you see that you can attach with the computer. You can put and load your sample over there, and then it sequences, and then those sequences are then stored in the computer, and you can do your data analysis over there. So it was assumed to be working well with these small genomes. Then there is a grid-on system. So here you have a set of cartridges put together in the shape of grids, and then you can increase the speed of reactions in parallel. So their claims are the read lengths expected is 48 KB. It's quite big. Throughput still not yet determined, and the reads in the test runs they were totally producing 2000 reads per reaction, and accuracy was low, and so far we missed data as far as the runtime is concerned, since it's in the phase of development. So different advantages which we can get from these three generation sequencers is that we need less sample preparations. We don't need to amplify them, so in this way we are safe against PCR errors. Same way we can have lower contamination, since pre-processing is low, so chances of having contaminations are also low, and no GC biasness as we see in the PCR's reactions. We can also amplify, since we don't need to amplify the fragments, so we can study any sample, even the smallest amount of the samples. We can have the exact quantities of the DNA or exact quantities of the RNA, since we are not amplifying them. So in this way, we might be safe against duplication errors, and we can sequence RNA directly by using these techniques.